The world of lab-grown organs. Imagine a world where no one dies waiting for an organ transplant. Imagine a failing heart no longer being a death sentence. Imagine damaged skin being replaced. Imagine kidneys that don't need donors. Imagine the human body being repaired piece by piece, not by chance, but by design. This is not science fiction. This is happening right now. This is happening inside laboratories around the world. This is driven by the revolutionary field of regenerative medicine, a discipline focused on rebuilding the human body itself. Step 1. Understanding stem cells. At the center of this transformation are stem cells. Stem cells are extraordinary cells capable of transforming into almost any type of human cell. This ability is called pluripotency. Heart cells beat. Brain cells process thoughts. Skin cells protect. Stem cells are blank blueprints. They can become any cell type in the human body. Induced pluripotent stem cells, or iPSCs, were discovered by Shinya Yamanaka. These cells allow scientists to take a patient's own cells and reprogram them. This eliminates the need for donors. It reduces the risk of rejection. Step 2. Collecting and reprogramming cells. The process begins with a small sample from the patient. Skin or blood cells are usually taken. Scientists introduce genetic factors. These factors deactivate the cell's original identity. They activate the cell's stem cell potential. The cell resets itself. It becomes capable of becoming any tissue. Step 3. Differentiation, giving cells instructions. Once reprogrammed, stem cells must be directed. They must become specific tissue types. Scientists control chemical signals and growth factors. They adjust environmental conditions and timing. Cells are guided to develop into heart cells, kidney cells, or skin cells. Step 4. Organ shaping, scaffolds, and 3D bioprinting. Cells alone are not enough. Organs need structure. Two methods are used: scaffolds, temporary frameworks guiding cell growth; 3D bioprinting. Cells are used as bio-ink, printed layer by layer into organ-like structures. This ensures cells form the correct architecture. It ensures correct tissue layers for organ function. Step 5. Bioreactors, growing the organ. The developing organ is placed in a bioreactor. A bioreactor mimics the human body. It provides oxygen, nutrients, and temperature control. Some bioreactors simulate physical movement. Heart tissues may be stimulated to beat. Muscles may be stretched to simulate growth. This helps tissues mature properly. Step 6. Mini-organs, organoids. Before creating full organs, scientists create organoids. Organoids are miniature versions of organs. They perform basic organ functions. They allow scientists to study diseases. They help test drugs. They allow refinement before attempting full organ growth. Step 7. Vascularization, building blood networks. One of the greatest challenges is vascularization. Blood vessels are needed to supply oxygen and nutrients. Without blood vessels, tissues cannot survive. Scientists are developing methods to grow blood vessel systems alongside organs. Step 8. Transplantation into humans. After development, lab-grown organs are tested. Tests measure stability, function, and safety. Successful transplants have already occurred. Bladders grown at Wake Forest Institute for Regenerative Medicine were implanted in patients. Lab-grown skin is used to heal burn victims. Beating heart cells and kidney organoids represent steps towards complex organs. Real-world impact and success stories. Bladders were rebuilt and transplanted successfully. They function long-term in patients. Lab-grown skin covers burns and regenerates real tissue. Heart and kidney organoids represent steps towards complex organs. Challenges and failures. Many experiments fail. Tissues sometimes collapse or develop improperly. Some transplanted tissues do not function as expected. The human body is extremely complex. It consists of nerves, chemical signaling, and blood networks. Replicating nature's precision remains difficult. Ethical considerations. Creating organs in labs raises questions. Are we playing God? Who will have access to these organs? Will they only be available to the wealthy? Could lab-grown organs be misused in black markets? These questions challenge ethics and humanity. Future possibilities. Lab-grown organs could eliminate organ failure. Damaged organs could be replaced, extending lifespan. Organs could be designed to perform better than natural ones. Brain-computer interfaces could integrate with lab-grown organs. Humans could be optimized with stronger immune systems and enhanced physical abilities. The philosophical questions. If we replace every organ, what defines identity? At what point does the human body become more machine than biology? Are we saving lives or fundamentally changing humanity? Powerful ending lines. The future is no longer coming. It is already here. Somewhere, a human organ is growing, silently, waiting to become part of someone's life. Humanity is learning to rebuild itself, cell by cell, layer by layer. With this power comes responsibility. One day, people may no longer fear disease, aging, or organ failure, but they may question what it truly means to be human. This is not just a story about science. It is a story about us. The question is no longer whether we can create life. The real question is what we will become when we do.